Hi, everyone. I wanted to welcome everybody. Thank you so much. And here comes Kate. Okay. Thank you so much, everybody, for coming. I know it's been a really difficult time for everyone, emotionally, physically, and for many people, financially. And so much appreciation for our speakers and panelists and their companies that are here today, and our moderators for all the wonderful work they're doing related to COVID-19. In addition to that, as we all know, there's a big movement around Black Lives Matter, and BVS is also very much in support of that initiative. And actually our next virtual event is going to be called Breaking the Barriers of Systemic Racism. In the past, we've done something called Women Breaking the Barriers of Science. And so we're going to do one on systemic racism and have different monthly virtual events. And so we look forward to seeing you at future ones. And thanks again for coming. Prasun, do you want to lead us off?
Thank you, Pam, and that's a great initiative, and looking forward to participating in that actually. Yeah, and thank you all for joining. I welcome you to San Diego's multi-faceted attack against COVID-19 pandemic, and the panel that will kick off is the Therapeutics and Vaccines perspective on COVID-19. And hi, my name is Prasun Mishra. I'm a founder and CEO of American Association for Precision Medicine, an organization focused on accelerating the field of precision medicine. And I'm also chair of ACT, which is AAPM's coronavirus task force. So the task force was launched right in mid-January, and we were one of the first global task force with over 200 members, in all continents, with a common dream to how to come together to accelerate drug discovery, diagnostics, as well as vaccine efforts to combat COVID crisis. And I have amazing panelists here with me. We have Erica and Saphire, who is leading the coronavirus task force. Hi, Erica. At La Jolla Institute of Immunology, we have Joe Payne, who is founder and president, he's a founder, president, and CEO of Arcturus, actually. And we have hi Joe. And we have Yvonne Snyder, who is a professor and director of the Center for Stem Cells and Regenerative Medicine at Sanford, Burnham. Hi, Yvonne, welcome. And we have Kate Broderick, who is a senior vice president R&D at Inovio Pharmaceuticals. Hi, Kate, welcome. So yeah, I think so. On the behalf of the organizing team, I would like to welcome and would like to just dive into one of the questions that we all have at the present moment, which is that with the recently WHO warned that coronavirus may never go away. So it's here to stay. So that's kind of a grim news. But so even when vaccines are here, the virus is here to stay for decades, and as it infects each and every individual, including children, which is very, very heartbreaking. So COVID-19 is like measles, HIV, chickenpox, and endemic actually, and stubbornly resisting efforts to stamp the disease. And some even have speculated that eventually everyone is going to know someone who got infected and died from the virus. So I would just invite your thoughts on these speculations, and how do we live with it safely going forward. Erica, I would start with you if you have.
Well, if the virus is here to stay, I think, and we can expect seasonal or annual return and some shift, I think it's just absolutely imperative that we have vaccines and therapeutics and diagnostics so that we can tell one respiratory virus from another. We see a lot of people right now that are quite convinced they had COVID-19 because they had some respiratory disease, but when they have an antibody test, they haven't. So very low current level of herd immunity means that we have a long way to go. And I think among us, we represent a variety of complementary approaches in developing vaccines to deliver protection, developing antibody therapies for those that can't be vaccinated or haven't yet been vaccinated, small molecule drugs to treat those that have been infected, and diagnostics to tell one disease from the other.
Thank you. Yeah. And Ivan, do you have any thoughts around that? Yeah.
I agree with Erica. I think that, well, it's interesting that coronavirus has been around forever. We knew it as the common cold. Every decade or so it flares up and its transmissibility diminishes and it kind of disappears. This one is severe. It has high transmissibility, and I agree, it will be here for keeps. But we're kind of used to this. I'm a pediatrician, and we just know that part of the battery of vaccinations that every kid gets is, and it's growing. Initially it was polio, and then it grew to MMR, which is mumps, measles, and rubella. And that just became fairly routine. And then we had influenza B, Haemophilus influenza, H flu. And then, of course, flu vaccines that we get every year. And it just becomes what we accept. And we're experiencing the badness of the unknown now. But I do think it'll be something that we're constantly aware of. And hopefully, it will just become one of the battery of vaccines, and then boosters, maybe yearly or every 5 yearly, depending on what we learn more about the virus that we just live with. But it is here to stay, like many other pathogens in our environment.
Oh, thank you. And Joseph?
Yeah, the only other comments I'd add is that I think we're getting more assurances or expectation that this will be a seasonal vaccine as well. It is a single-stranded, self-replicating mRNA payload inside of that virus, and whenever you have this type of virus with a simple, an RNA vaccine, that these respond to temperature and humidity, and that's a seasonal aspect. But how long it will take for that seasonal pattern to engage, of course, is up in the air right now. We're definitely in pandemic mode still, and we're talking about first and second waves. So we're getting ahead of ourselves to call it a seasonal vaccine, but that's the expectation as well. It's here to stay. It will fall in line with the seasons, and then hopefully we'll be able to address it as good as we have with the flu, and potentially a lot better as a group of scientists and in the medical community.
Thank you. And Kate, any thoughts around
Yeah, as I say, sobering thought, isn't it, that it's here to stay, but the data certainly points in that direction. As scientists and clinicians, that's a daunting thought, but let's imagine what it's like for the general public too. This is a very confusing, frustrating time for them. And I think, as Eric rightly pointed out, we need to be vocal about the potential vaccines, therapeutics, diagnostic tests, and really it will be the credibility and the strength of those three arms of attack that will allow us to kind of consider this hopefully like a seasonal flu vaccine. And of course that depends on the duration that the vaccine's able to deliver. But we're all in that area, work very hard to ensure that it's safe, effective, and durable.
Yeah, I think that's the, so I think I was hoping that there will be some resentment that the virus is not here to stay, but I think we got a we got a unanimous I think vote that the virus is going to stay, and that's what my feeling is, and that's kind of would drive the whole conversation from now onwards with how do we, and I think some of you pointed out how do we live with it safely, right? So I think so. The question is, so one of the, I lead an organization, is American Association of Precision Medicine. So I always worry about, how we can not worry, but how optimistic how we can utilize precision medicine-based approaches. So are there any gender and racial and gender susceptibility to COVID-19, and are these disparities well addressed in the clinical trials? Actually, can anybody would like to take this?
Could start if I could, and certainly
sure, yeah, go ahead. Yeah,
it's been very well covered in the media that COVID-19 disproportionately affects people in minority communities and also those with comorbidities. I think the early phase 1 trials that we're all aware of for vaccines tend to target, of course, a very small portion of population demographics. But as we roll out the phase 2s, the phase 3s, certainly I can speak on behalf of Inovio, we're looking at extending into a much more diverse population so that we're able to assess the vaccine efficacy across all demographic groups, ages, BMI types, and some potential comorbidities. And I do think that's absolutely vital as we ask the important questions of efficacy, that the trials are designed in a way to be inclusive and reflective of the actual population.
Any other takers of this question?
I may be able to weigh in a little bit on this. And
okay.
So it may be odd that that a stem cell biologist should be on a panel about viral diseases and inflammatory diseases. So this is actually where I think we can contribute, and this is kind of how I became involved in this. So one of the areas that stem cell biology has entered is modeling diseases. So everybody's familiar with the aspect of stem cell biology where you take a stem cell and you stick it into something bad, hoping for something good. That's one area of stem cell biology. The other area, and this actually gets into your area, Prasun, is modeling diseases, but modeling diseases in a patient-specific manner. So this is something that our group has been doing now for a number of years. And the way one models diseases, well, it goes back to the earliest days of the stem cell field, where we knew that we could pull a stem cell out of the brain or the bone marrow of the heart. And typically, especially for the brain, we would need to wait for a patient to die. And if they had Alzheimer's or Parkinson's or some other disease, we would grow their cells. In 2007, as everybody knows, Yamanaka recognized that he could take mature, very accessible cells from the skin, from the blood, from the hair follicles, and reprogram them back to as if they were very immature cells just in the embryo, and then move forward and make them become brain or blood or heart or pancreas or whatever one wanted. But interestingly, they had the profile, they had the genetic fingerprint very, very often of the patient from whom those cells came. So the conceit was you had a neural stem cell and then a neuron from it, but it had Alzheimer's disease or it had Parkinson's disease. And as we started exploring, it actually acted like it had psychiatric disorders like schizophrenia or bipolar disease. And then it became known that you even got a more representative picture if you didn't just grow these cells in a single two-dimensional monolayer, but you actually tried to create a mini organ in the dish. So a mini brain, a mini heart. And purely by happenstance, because my area of pediatrics is neonatology, which heavily deals with lung disease, I had a fellow neonatologist in my lab looking at the lung diseases that babies get. And her name is Sandra Leibel. And part of my mission, even though I'm trained actually as a neuroscientist, though clinically as a neonatologist, I deal with all systems. So I was exceedingly welcoming about reaching out, and I try to do this with all physicians, to bring them into the lab to do science. She brought her interest in lungs to the lab and started making mini lungs in a dish, lung organoids, looking mostly at the diseases that babies get. But it became obvious that when she was putting these together, she had the only authentic mini lungs in the literature that had all the cell types, that had the cells that made surfactant, that had the cells that had cilia. And then we could actually add other aspects to the lung, like macrophages and blood vessels. We were actually thinking, what area could we use these for other than just babies' diseases? And COVID-19 hit. All these questions came up, like, we think it hits the lung, but what cell type in the lung? Are the patients really dying of what we call adult respiratory distress syndrome or acute respiratory distress syndrome, which is a loss of surfactant, which is, as a baby doctor, we deal with all the time. Is it pulmonary edema? What cell type is it? Is it hitting the macrophages directly, or are those just unfortunate bystanders and things of that? And we decided we could answer that question. We could start infecting the lung organoids, either with a full virus or a pseudovirus. But then it started becoming evident that not everybody was getting the disease with the same severity. So the first aspect that that people started recognizing was that somehow kids were not getting a pulmonary disease the way adults were. Well, anybody who works with stem cells knows that you start off with immature versions of an organ, and then it matures in front of your eyes. And we started seeing that the young lungs don't get infected. Only after they become mature do they start becoming infected. And then it started becoming out that African Americans and Latinos were getting the disease worse than other racial groups, and that men were getting it worse than females. And we realized we could answer that question. Because remember I said, you take the cells from the patient, and now you have patient-specific organoids. So now we're starting to make African-American organoids and Latino organoids and male organoids and female organoids. And we can answer the question, hopefully, is it that, that certain racial groups also suffer from adverse socioeconomic circumstances? Or is it the presence of comorbidities? We can recreate the milieu. Or do they have just a different HLA type that makes them more susceptible? Or are the cells themselves actually different? So one, that's an interesting scientific question to ask. And hopefully what we're going to do is find out that there's nothing fundamentally different. It really is adverse socioeconomic circumstances, which hopefully we can address. But even if we can't answer the question, at least as we discover new drugs, we can make sure that altering the dose or altering the drug regime should, if we hit a good drug, cover all patients, everybody who suffers from this disease. So I think that's how a stem cell biologist can contribute to this question of disease disparity.
No, that's a very good input, and I think there is on therapeutic side there is also folks have used mesenchymal stem cells for to treat fibrosis. So I think it is really an up and coming area, and thank you for sharing that perspective. So I will dive into now the specific question, and first question I think to you, Joe, will be in your opinion, what are some of the leading vaccine candidates, and how does the Arcturus vaccine compares to Moderna? Actually?
Sure. So I think Moderna and Inovio have gotten a lot of attention because of their first mover capabilities and execution, and rightly so. And we have some from Inovio on the panel here today, and we'll allow her to speak on behalf of Inovio. Moderna is a messenger RNA vaccine. So is the Arcturus approach that people are monitoring and watching closely. I think Novavax is getting a lot of attention from the protein subunit world, along with their adjuvant they have an adjuvant approach, and then there's others, and I'd say internationally, I think Oxford and AstraZeneca are making a lot of press right now. But how is Arcturus different is the key question. Even though we're a messenger RNA vaccine, our mechanism is a self-replicating messenger RNA vaccine. When a messenger RNA molecule goes into a cell, it expresses the S antigen, and then it degrades, and it requires a booster shot. So the self-replicating mRNA mechanism has an advantage in that once it enters the cell, it not only prints off the S antigen, but it prints off an RNA-dependent RNA polymerase, which enables the replicating aspect of it. So the kinetics of this means that ultimately the expression of the S antigen lasts for 2 to 3 weeks instead of 2 to 3 days like a conventional mRNA. So when we initially approached this vaccine, we looked at both conventional mRNA, our internal efforts on conventional mRNA and self-replicating mRNA, and what we came to realize, self-replicating mRNA works at much lower doses and is a potential single shot, which is a significant advantage as well. And when I mean lower doses, I would say that Moderna has locked in their dose for phase 3 at 100 micrograms, and 2 of those doses, and we're looking at much lower doses than that. In fact, in our preclinical animal models, in our animal studies, we saw 100% seroconversion at 2 micrograms. And we saw 80% seroconversion at 0.2 micrograms, a single shot. So that's where it becomes interesting, and then that's the key differentiator. It's a potential single shot, and it's a very low dose vaccine, and those are important for logistically distributing the vaccine. As we've engaged all these countries, you can imagine the complexity of administering a multi-dose vaccine versus a single shot. And then from a manufacturing efficiency and cost perspective, you can imagine the difference between making 50 metaphorical factories versus 1, or 50 people versus 1. So the cost and then the speed, I'm not saying we'd be 50 times faster, but we'd definitely be more efficient. Absolutely. So there's advantages to that. Did I address your question on how we're?
Yeah, I think you did. So I think I would, as you invited, I would invite, Kate, I think, would you like to share how, how your approach is, similar or different than the approaches that Joe mentioned?
So Inovio specializes in DNA-based approaches. So our vaccine for COVID-19 is a DNA plasmid, so a circular piece of DNA. So as such, it's extremely stable, which is a real benefit. We don't require frozen cold chain, so that's one differentiator against quite a lot of different conventional vaccine approaches, and also other nucleic acid approaches. One of the important aspects, though, I think about DNA vaccines in particular is the breadth, I think, of the immune with this approach. And certainly, of course, we're doing that right now. Phase, we'll hope to with our effort forward. When you cast your mind historically back to our infectious disease program, so for last HIV, Merck, being able to drive a very cell response, which I think is fairly standard for vaccine approaches, but I think we differ in the fact that to T cell responses, and I think more and more as you go through the literature, that those T cell aspects will be really quite important. We're thinking about those T cell, and just one last safety profile of those DNA is really, really very, very benign in COVID-19. So that we've reported all have just been grade 1 and are, injection site reactions. So very, very standard approaches. And that's, I think, somewhere where we differentiate ourselves from some of the other vaccine platforms.
Thank you for sharing that. In fact, that was my next question to you. But I think that thank you for clarifying. And then overall, Joe and Kate, I think I would like to bring this point to both of you that as we have learned from the flu vaccine, that the vaccines are marginally efficient. Although there is a very high bar for the approval of the vaccine, but I think the virus mutates. So what do you think, in your opinion, how rapidly the SARS-CoV-2 virus is mutating, and how safe, effective the vaccines would be actually, and will the vaccine give us a hard immunity against COVID-19? So I think these are multiple questions, but all linked.
Yeah,
so Joe,
early in the stages, we evaluated a lot of different types of vaccines. We evaluated the full-length spike protein and expressing that, or truncated portions of it. And we ultimately decided on doing the full-length spike protein for many reasons, and we're very happy we've done that. But one of the advantages of expressing the full-length spike protein in its native conformation is that if there's a mutation way over here, we generate a family of antibodies that are neutralizing to the full-length spike protein. So are we expecting mutations to occur? Absolutely. Are we concerned about it? Right now, no, because in theory, a robust theory, that the patient or the individual, the participant in a trial, will be still protected with neutralizing antibodies and other portions of the full-length spike protein. I think it makes sense. If there's a little tiny mutation over here, it won't impact any neutralizing antibodies over there. And that's our approach. And that's actually a common, it's not differentiating. A lot of the vaccines out there are pursuing a full-length spike protein in its native conformation type approach.
I'm actually looking at that question.
All right, got you, Kate.
I'm the director of an immunotherapy consortium that's looking at the several hundred molecules being raised for therapeutic antibody treatment around the world, and we're mapping, correlating them to the minute changes in spike to see which individual antibodies are knocked out. On the polyclonal level from a vaccine, I wouldn't expect that much of a difference, but we are looking to see which types of individual monoclonals will be knocked out.
Okay.
Yeah.
Got you. Kate, your opinion on opinion on those 2 points raised, and will a vaccine give us a herd immunity against COVID actually, which is now endemic?
Great questions, and certainly from kind of the inception of our development program, we've been keeping a very watchful eye, as I'm sure all of my fellow panelists have been, on the the mutation capability of this virus. Mutations, it can go 3 ways, right? It can be less infective, more infective, or kind of nothing. So, I think we should all be heartened to know that there doesn't seem to be any more potent strains around, which is a positive thing. And certainly we're not seeing the mutation rate that you see for the flu or HIV. And that's really been covered a lot in the media as, well, what's our hope for COVID if we haven't, 30 years on got a vaccine for HIV? And I do think it's important to differentiate that they're really very, very distinct viruses. So, very much to Joel's explanation too. We also use the full length spike protein, and for a number of reasons think that's really the best approach for a vaccine here. In regards to will a vaccine be able to achieve herd immunity? We hope so, but to be perfectly honest, and maybe my fellow panelists will agree with this, Prasun, there's still so much we don't know about this virus and about this disease, and we're really still in the learning stages as much as everyone else.
Thank you for sharing that, and I feel the same. The feelings are mutual, actually. So, and that brings us to Erica. Erica, I think you have led an international, COVID-19 task force at the, at La Jolla Institute for Immunology. So your learnings of, how institutions, groups are coming together and collaborations being, formed in the field of diagnostics, therapeutics, and vaccines. All your learnings that you have, if you can share some of those with us. How was it to lead this, and then what what were the accomplishments and where you were going?
Still in the thick of it. I think the important thing to notice is San Diego is a whole lot more than fish tacos and tan thighs. That there's a lot of science here, and we're the headquarters for the global collaboration to try to figure out which of the hundreds of excellent therapeutic antibody molecules going forward are the most potent, what makes them potent, what are the best combinations in which you can get additive or synergistic effects. And then the focus of the Gates-funded effort in my lab is to figure out which of these can be mobilized to low and middle income countries, which antibody therapeutics are most potent for lowest cost of goods, so they can be distributed broadly and maybe distributed without needing IV delivery. And so that has involved a lot of careful thought and assembly of how you get 50 to 70 different groups, from academic labs to biotechs to multinational corporations, to pool samples and intellectual resources. And we're doing it in an anonymized, blinded fashion where we don't know what molecule is named what, but we're gathering a broader, deeper data set than would be possible with any group alone to figure out what do antibodies do, which ones behave in which way. And then from that, NIH and Gates and other organizations can make therapeutic recommendations.
Wonderful. So I think it looks like this is an excellent effort, and we look forward to more coming out of it. So I thought Kate, you'll be best to or Kate or Erica, maybe if you can comment on the convalescent plasma antibodies. Erica, maybe you think and hyperimmune globin therapy, and other advanced therapies that are that are being used so that we also touch upon those topics.
These are important proof of concepts
for COVID-19,
and you have convalescent plasma in existence before you have mobilized and tested individual monoclonals, and so that's a good proof of concept. It's harder to scale things which have come out of people, and there's quite a lot of variability. Some people make a very potent neutralizing response, others don't. With animal hyperimmune sera, you worry about a reaction against that. Those are used fairly frequently in low and middle income countries against things like diphtheria and antivenom. But we have such potent monoclonals that are fully human that I think we'll be able to deliver a fully human product.
Okay, so you think that antibodies, themselves will be sufficient? We don't need to inject the plasma. That's a
highly potent, well-characterized monoclonal antibodies evaluated for safety and efficacy will be a better product to put into people than more poorly characterized polyclonal. They can certainly be scaled up to a greater level. Now, that's a theory, right? We have to see the clinical data, but that's my expectation.
And your opinion on how is it working, the convalescent plasma, the clinical trials, I think it's spearheaded by Mayo Clinic, but how can you comment on the results?
No, I'm not intimately familiar with those.
Are they?
None of the other panelists are.
Anyone else has a insight on that? If not, then I think we can move on. But I think this is one of the efforts, and there were some reports that some patients were responding. So I think that's one good therapeutic modality that's out there, and that's one effort that's
Prasun, could I just
ongoing.
add
So,
just something to that? I think might be of interest to the listeners.
Sure, wonderful.
Certainly, I can't comment on the trials, but certainly what I can comment on is the massive variability that we see when we're looking at convalescent patient samples. And I think that, generally, of course, in a vaccine development protocol, you use those as kind of a gold standard. If someone's got better from the disease, then this is kind of the areas of the immune response that you want to home in on. However, certainly it's something that we're seeing with COVID, and Erica, I'm sure, can comment much more deeply than I can on this, is the huge level of variability that we're seeing in neutralization titers, binding antibody titers, that kind of thing. So I think it is maybe important for people to understand that although we've called it COVID-19, really is quite a spectrum of different elements under that very broad umbrella.
Got you. That's a very valuable insight. And with that, I think, Evan, I think we'll come to you. And so I think, so although you are a stem cell, biologist, right? And we are very hopeful for the stem cell therapies to, post-COVID interventions. But, I think Sanford Burnham is also known for repurposing of the drugs, right? So I just, as a leader, I thought, I think if you can share your perspective on, how, what are the efforts that are that are ongoing to repurpose the drugs for COVID-19, which will be the fastest way to do it. So any success stories, examples that you can share us all?
Sure.
That will be great.
As you said, I think our institute is known for two major things. One, for stem cell biology, and the second is for assay development and drug discovery. And what's very interesting is that I mentioned that a big effort in the stem cell field now is modeling diseases. One of the reasons for modeling diseases is not just to figure out mechanisms, but to discover drug targets, and then to actually use the cells to discover drugs against those targets. Initially, completely independently, Sumit Chanda at our group, who is a virologist and drug discoverer and assay developer, did this amazingly heroic task. I think you may have heard about it, where early on in January, he and his group, in collaboration with the group in Hong Kong, screened 12,000 FDA approved or late stage or clinical stage drugs to try to find something that would block the cytopathic effects of the virus. And he did that. And I won't tell the whole story the way he tells it, but of 12,000 late stage drugs, and of course the advantage of doing late stage drugs is that they've already made it through most of the regulatory gauntlet. Of the 12,000, he found 30 that looked as if they blocked the cytopathic effects. Of the 30, 6 looked as if they could actually achieve the EC50 for those drugs was one that could be achieved fairly readily in patients. And the one thing was that, as many of you know, the cells that one needs to do for high throughput screening, particularly for antivirals, are very unrelated to the clinical situation. They're various cells which are monkey kidney cells, or human carcinoma cells, or human hepatocarcinoma cells. So that's where we came in. To help winnow the list and fast forward it, we are now using lung organoids to figure out which of those 30, and ideally which of those 6 that looked as if they were very effective in blocking the cytopathic effects could actually work in a clinically relevant system, again, taking into account all kinds of disease disparity, what we've decided to focus on, there are 6 that are useful. We're focusing on some both narrow and broad spectrum protease inhibitors. And it's often been said that the proteases might be the Achilles heel for this virus. So obviously the vaccine, that is nirvana. Okay. And to get there, drugs would be useful.
Okay. Wonderful. And then I think you were using even the organoid models as well as the screening. So what would be the ideal repurposing candidate, using the approaches that that are being used? That's
So what we do is we take the organoids. They can be infected both with a pseudovirus. It can be done in medium to high throughput in 96-well format. The pseudovirus has a luciferase conjugate. It can be read by a plate reader. We also are doing the full virus, which can also be assessed based on antibodies against the nuclear protein or PCR. And we are just screening drugs that look as if at the dose could be fast-tracked through the regular. We probably don't need to do animal studies. Most of these have already been in animal studies. We know their safety, we know their dose, biodistribution, CMC, the whole bit. We just don't know whether they work in an actual lung model. We'll get those answers and then hopefully fast track through the FDA and start clinical trials in a few months. As I mentioned, some are unexpected. They work for unclear reasons. Some are more expected, like the protease inhibitors. Some are other antimicrobials that can be repurposed for this reason. So I think we're making progress. I'm fairly hopeful that before the end of this calendar year, we'll have something that can go into clinical trials.
Wonderful. Oh, that's a good news, actually. Yeah, so I think we have, it's amazing discussion, and the momentum is going. I think this warrants the whole day call. Let's call each other and discuss. But I think we have a limited time. So with that, what I will do is I will just ask, overarching question. So if we were to look at the COVID-19 pandemic as a case study, so far whatever we have learned, how can we do better in the future? And any concluding thoughts or call for action?
Better
Erica, I'll
understanding
start with
correlates
you.
of immunity and broadly applicable platforms that can be pivoted rapidly.
I would suggest we should be more prepared. I think the entire world has learned a lesson. If we had a massive earthquake, the same sequence of events would have happened. But back in 2015, the WHO was warning all the countries in the world to begin stockpiling for COVID and many other pathogens, and no one did, or at least mostly. And I think the world's learned our lessons. So I think one of the key take-home lessons from this is that we not only address this pandemic as it enters into the endemic phase, but that we take a mature look and actually budget for stockpiling vaccines of all their pathogens, so that we can nip this in the bud next time that one of these pathogens rears its ugly head. We can immediately deploy the vaccines and just quelch it in the early days.
I think I would, I feel quite strongly about this, and I'm sure my colleagues here do too. In the past, we've taken a very reactive approach to vaccine and therapeutic development, and clearly this has been a perfect demonstration of how that was not the best scenario. So I do hope that we can, when we all take a breather, reflect on the situation and say that is not the best way for vaccine development or other developments to be carried out. And lastly, I would say surveillance of viruses in animal populations, because this will happen again, and we should be very, very aware of that.
Yeah, I agree with
Evan?
all of those comments, and I think stronger federal leadership would help to be looking at the science, trusting the scientists, and preparation and organization would have made a huge difference here.
Thank you very much. And I think that brings us, we are right on time, and we're right on time. And I think, so together, I think what we have learned is that, importance of agility in drug discovery, vaccine development, and the whole the COVID-19 pandemic has taught us the importance of collaboration, working together. I think we can bring, we can in a small time frame, we can, as we work together, we can we can make a greater progress. So I think and thank you for sharing all your thoughts and insights and experiences. And with that, I would conclude this panel. Great discussion. Thank you everyone, and thanks.
Thank you.
Yeah, so thank you to everybody. And you know what, there was quite a number of questions in the chat box, and Andrea, who works with my team, is going to capture those, and maybe I could send it off to the speakers, and then we can get some answers to that, because I know we had a lot to cover in the panel. So
thank you. That's a great idea.
Yeah.
Thank you for doing that.
Thank you so much, everyone. If our next panelist